Hi, everybody. This is Dr. Wiseman. I'm your instructor for Bio 330, Biomedical Physiology. My contact information is here. You can text me or call me anytime in these hours at this number. In fact, this may be the easiest way to get a hold of me during the day if I'm teaching other class or if I'm doing something else. You can also email me as well. I'm glad to hear from you. And this semester, these are my office hours, my standard office hours, LSH 110E, which is down the right-hand hall, just past the biology lobby there, from 3 to 5 Tuesday and Thursday. But I do have some flexibility, and I'm happy to meet you by appointment if you let me know ahead of time. I'll be glad to make time for you, even if it has to be on a weekend or something, if possible. So here's my standard teaching schedule. You can see that I have a morning section and an afternoon section of 3:30. Here are my office hours, 3 to 5, Tuesday through Thursday. Not available in the evening on Mondays or Wednesdays. But generally anywhere else where you see white is theoretically possible. Trying to leave a lunch hour in here in the middle, especially between classes, so that I can kind of keep the blood sugar up, if you know what I mean. One thing this semester that I'm going to emphasize a little more than I have in previous semesters is that you refer to bright space before you go too far. Rather than just ask a question, I would ask that you first check bright space to see if the information isn't there first. I have the entire semester calendar plugged into the Brightspace calendar. I'm also going to be publishing a visual calendar as well. This is also where you're going to find things like resources, like, for instance, if you want to take in a lecture or something along those lines asynchronously, we do have 1 week this semester that's going to be asynchronous. I've got the links there, weekly content as well as announcements and so forth. I'm sure you're familiar with Brightspace, but I wanted to just reinforce the idea that a lot of the questions I often get can be found relatively easily and quickly in Brightspace. Sometimes I'll say in class, welcome to your first day of physical therapy school or welcome to your first day of medical school. That's because physiology is your guide star to what it is you're going to be trying to achieve in your patients or clients. That is to say, we have mechanisms that are in play that will keep an organism in a state of what we call homeostasis. That's not static. That's one of the problems with homeostasis is that it implies that there is no movement, and that's anything but true. All right. Homeostasis is dynamic. It's what they call a dynamic steady state, and things are changing all the time. And that's because the environment changes, the situations change, the metabolic needs change, and so forth. And that's what we show here is that you could have an external change, like, for instance, maybe you walk into a walk-in freezer, all right, or maybe you jump into a swimming pool, or perhaps you're feeling stressed because you've got an exam next hour. Okay, these changes, whether they're internal or external, lead to a departure from what we call a homeostatic window. And I'm going to be reinforcing that concept all semester. This idea that homeostasis isn't any one value. Like, for instance, we talk about body temperature, 98.6 degrees Fahrenheit, prefer to be in Celsius, which is 37 degrees Celsius. Well, our body temperature is fluctuating all the time, whether you've just had a meal or you just woke up or you've been asleep, or you've been working out. Your body temperature changes, and it's a range of body temperatures over the course of a day. It's not any one. And so we have this homeostatic window, and if we have a change of some kind, we may depart outside of that normal range of controlled values. Now, what will generally happen is that the organism, the person, the animal, or so forth, it's a cell, will attempt to compensate. And if it's able to compensate and bring the homeostatic controlled value back into the proper range, that's what we call wellness. However, if we depart from that, that's what leads to what we call illness, disease, injury in some cases. All right. So Homeostasis and the physiologic mechanisms that sustain homeostasis are your guide star, if you will. They are the standards by which you will be attempting to achieve wellness in your patients. And let me show you an example of why it's important that you have a diverse understanding of physiology, not just maybe a relatively focused situation. Let's say you're interested in physical therapy for athletes, like you want to be a trainer for a professional football team or something. Then that's relatively narrow range. You have relatively young patients, they're in good shape. Most of what you're going to be dealing with are injuries. All right. Well, perhaps one injury is something that we would call exertional rhabdomyolysis. This is the condition where if you have some kind of extreme exertion, and it can even be not even exertional. Severe exertion is one thing, trauma can do another, heat injury can also occur. What's happening in rhabdomyolysis is the muscle fibers are literally breaking down and releasing their contents into the extracellular fluid, which leads to potential for kidney damage and heart arrhythmias, and all sorts of other sequelae. That's because while muscles are maybe the point of emphasis for someone involved in physical therapy or occupational therapy and so forth, nevertheless, the muscles are intimately related to the rest of the body in terms of metabolic inputs, chemistry, blood pH, blood constituency. And if you have enough rhabdomyolysis occurring, what will happen is the myoglobin that's found in a lot of skeletal muscle, it's a close cousin of hemoglobin, will get out in and it will hang up in the kidneys, either as myoglobin or as myoglobin metabolites that lead to kidney failure. Acute kidney failure is not uncommon for someone with severe rhabdomyolysis. You can see lesser severe situations, like for instance, if they've worked out very, very hard, they ran a marathon or something the next day, the urine is much darker, often a red or a brownish tint to it. I had this happen to me personally. I was caving in a cave in New Mexico where we were going down for a week. And the temperature of the cave was warm enough and it was humid enough and the work was hard enough that I was actually experiencing a little bit of an issue in the cave, and I had to be very, very careful that I didn't get any worse, or I would have had to have left. That's the connections that we're dealing with. We're dealing with lots of interconnected pieces. You may specialize in muscles, you may specialize in fitness and exercise, but that doesn't absolve you of the responsibility for knowing the whole body. Even in physical therapy itself, there are so many different kinds. Here, just in this picture alone, we have 8 different types of physical therapy, each with a different emphasis, using different apparatus or different approaches for handling whatever it is you're attempting to achieve in these patients, whether it's wellness, improved fitness, rehabilitation, and so forth. I'm going to be giving you guys a couple of articles about this again to remind you that it's not enough just to have a handle on exercise physiology or basic physiology. You have to understand physiology at the level of therapy, and that includes drug therapy as well as manipulative and massage and those other things. And this is just a figure showing 6 of the most common things that physicians treat: diabetes, hypertension, issues with breathing, cardiovascular issues, GI disturbances, liver, and so forth. Those kind of be lumped together a little bit. These are the common ones. You have to not only be ready for the common ones, you also have to be ready for the slightly uncommon and even the rare. And that's the point of starting with physiology first. Sometimes when we talk in physiology, it feels very far away from the kind of care that you want to give or that you're particularly interested in. Just like an astronaut might be interested in flying and going out into space, still has to know about physiology to understand how their body's going to behave in zero gravity or with reduced oxygen and pressures and G forces and so forth. You have to have a diverse and rigorous education in order to be fully prepared for the clinical possibilities that you will be faced with as caregivers. So the course is broken up into 4 major units. Some students will ask me, is the course cumulative? And the short answer is yes, but with some caveats. The main thing I would say is that Unit 1 is the cumulative part of this course. That is to say, the elements that we're going to study in Unit 1 apply to all of the other units, and we're going to be returning to those themes and those mechanisms throughout the semester. So we're going to be dealing with a little bit of biochemistry, so protein function and so forth, membrane function, acids and bases, osmolarity and tonicity. These are things you've probably seen before in things like Bio 165, but we're going to apply them specifically to function, to physiology. We'll finish off the unit with a little bit of cell signaling, and this is a connector chapter for Unit 2, which is signaling physiology. But rather being intracellular signaling, which is what this picture is showing you here, we're going to be dealing with long distance signaling, things like the endocrine system, the autonomic system, rest and digest, fight and flight, neurons. We will also talk about the sensory things. We have somatic senses and we have special senses. All of this, again, is cycling back to the basic cellular physiology. And I have a synapse here because, again, a lot of what we're dealing with is going to be at the cellular level. Unit 3 is contraction physiology. So again, we're going to apply some of the principles that we see previously. Now we're going to apply them to muscles, like for instance, skeletal muscles and cardiac muscle. We'll talk about electrocardiograms and smooth muscle. So Unit 3 is fundamentally about how these contractile cells are stimulated and how they respond, because they don't always respond in the exact same way. And a lot of what you guys deal with as therapists and clinicians involves muscular function, whether it's skeletal muscle, loss of function, injury, or it's exercise intolerance, cardiac muscle, or perhaps some other type of disorder that involves smooth muscle, like a digestive problem or some other sort of thing. So we're going to go into the muscles in this semester and how the muscles get the job done. Finally, transport physiology, and this involves things like breathing, respiration, gas exchange, how we move oxygen and carbon dioxide, and lastly, how the kidney does its thing. The interesting thing is that Unit 4 in many ways cycles back to Unit 1. A lot of the processes that we will talk about here are applications of Unit 1, and sometimes these other ones, into these systems of respiration and renal function. So is the course cumulative? Yes. Not specifically cumulative, but certainly we are going to be using, it's scaffolded in that way. We're going to be using a particular Unit 1 in Unit 2 and so forth and so forth. For Chapter 1, we have the reading assignment pages 1 through 22, and we're going to be going through some very, very general themes. Now, don't blow this off, because these themes help you to recognize patterns. And if you can recognize patterns, then if you're not entirely familiar with that system, at least you could understand the basic physiologic goals. A lot of times this will feel like hand waving. It will feel very abstract, or, okay, I already get that kind of thing. Don't leave it behind, because there will be times when you will not be completely familiar with something. And then you have to kind of start from scratch. Well, you start with the basic physiologic goals and the basic themes, and then things will begin to make sense rather than being completely arbitrary, which is what memorization is very weak at. It's very weak at trying to remember something absent of context. And this class is all about context. So when we say integrative science, what we mean is we're going to be integrating chemistry and physics and cell biology, which is already chemistry and physics, to help you understand what the physiologic goals are and infer what function may be. And that leads us into function and mechanism. Again, mechanisms are going to be at the cellular level. If you've had Bio 104 or you've had other types of physiology courses, those are not going to be sufficient to get through many of the mechanisms that we're going to be covering because we are going to be at the cellular level. Homeostasis, we all kind of know what homeostasis is. We're going to break that out. And again, the idea is when in doubt, what makes sense from a homeostatic level? Now we often say, oh, it's a dynamic steady state. Well, what does that mean? It means that we're trying to keep things in a given range. It's too much is bad, too little is bad, and we're working all the time to keep us in that what we call homeostatic window. So when I give lectures this semester, I'm often going to talk about this idea of homeostatic window and how we're trying to get back to a given or sometimes go away from a given homeostatic regulated set point. We are going to deal with energy flow a little bit, not a ton. Chapter 4 is where most of that's going to happen is energy flow, but we still have to kind of touch on it a little bit in order to set you up for more advanced classes down the line. If you think about it, energy flow is exceptionally important when you think about things like weight loss, okay, or the ability to contract or relax and so forth. And so we will see elements of energy flow, though it's not one of the primary themes of chapter of Unit 1. We're going to just touch on some of that stuff. Control systems, on the other hand, extremely important. So we're going to emphasize control systems, and what we mean by that are basic control systems, whether it's negative feedback or positive feedback, feed forward, etc. Again, when you encounter situations where you are not fully familiar, how do we get back to basics so that we can make informed decisions for our patients? And it's coming back to control systems, homeostasis, understanding of function and mechanism. So when we say physiology is an integrative science, what does that actually mean? Well, crossing boundaries. We're going to be merging things like chemistry, acids and bases, energy flow through chemical means, cell biology processes, using cells in order to achieve larger, shall we say, tissue level and system level goals. Certainly some physics in there. We're plugging all of these things in order to understand how it is that the body gets the job done. Sometimes the body gets the job done by maintaining a given set of regulated variables, like for instance, body temperature or blood pH or so forth. There are also times when we need to depart from those physiologic set points in certain situations. Let me give you an example. A sneeze. You would not be able to survive if you were sneezing perpetually. But a sneeze does have a physiologic justification, but we can't use it all the time. So there are going to be times when we're going to be trying to bring it back to homeostasis, and there are going to be times when we're going to depart from homeostasis. And that's what this class is about, is to help you understand the reasons why tissues and cells do what they do. Physiology is fundamentally a conceptual course. That is, we're going to be talking about concepts and talking about mechanisms. Well, for starters, that means terminology. As you go through the book, my recommendation for you, if you want to be as successful as you can be, is note where you see bolded and where you see italicized. This means this is top level information. I do see where students have more difficulty is when they are not, the word is discrimination, when they are not discriminating between what is really important stuff and what is not so important. And so in this course, one of the things you're going to want to do is identify the really critical stuff. That's your high calorie stuff. That's where you're going to get most of your points, is where you see bolded and italicized phrasing in the textbook. So learn those terms, understand what those terms mean, be able to use them rather than a recitative way, but a conversational way. We are learning a new language here. The other thing about physiology and advanced clinical understanding is that there is a lot of it out there. Just like we don't memorize the dictionary, we learn how to use the dictionary as a tool. Physiology is sort of like that for the human body. It gives us some things that we can use when we're faced with things that we are not entirely familiar with. So it's a new approach rather than being told exactly what is or what isn't. There's a fair amount of that in this course. We are also helping to try to prepare you for the idea of discretionary thinking. Critical thinking is what we call it in the liberal arts school, where you have context in order to make decisions. So it's a new approach. Yes, we're going to talk about big ideas like homeostasis, but we are going to drill down into mechanisms and interactions. You can understand homeostasis, but not understand how the mechanism keeps it there. We are going to be very, very down in this level throughout the semester. Mechanisms, and we say interactions, that's what we mean by biomolecules, cell structures interacting with each other in order to convey energy or convey information. So this is a very mechanistic course. One of the challenging aspects of being what we call an integrative science is that we have many levels of organization. We have the molecular or even the atomic level in some cases. We have the cellular level. We have tissue level and the organ level and system level. That can get really complicated really quickly because when we have multiple levels and multiple inputs, oftentimes the behavior of that system cannot be predicted by the sum of its parts. That is to say, our bodies are carbon and hydrogen and phosphorus and oxygen and so forth, but we are not just a pile of elements. We are complex systems. And that complexity is often described as an emergent property. So what are some examples of emergent properties? I have a couple of videos here and I strongly encourage you to check them out. I think they're pretty cool. Like for instance, if we look at this system right here, it's actually a couple of wheels, one of which is hooked to some sort of a powertrain of some kind. You can certainly imagine what would happen if you were to put a person on this bike and so forth. But would you have predicted that that system could be behaving in this mechanism? It's called a flat top maneuver here. That's an example of an emergent property that even though we still have two wheels and a drive chain and so forth, nevertheless, if we add additional inputs, the complexity arises quite quickly. And that's the challenge of physiology. We add one variable and now the system is much more complex than it may seem initially. Here's an example of emergent properties. And again, this sort of ties into how we're going to try to explain some of these processes to you. When we look at mechanisms, we will often break them down into sort of single point sorts of things. You can see that this animation here, we have one ball being tossed. So that makes sense and it's easy. But when we start applying balls, now we begin to see a differential behavior going on. Again, if you'll notice, the humanoid here is still catching and tossing the ball, whether it's one way or another. Here's an example of an emergent property here. Now, if we add even more balls, now it's getting kind of complicated. Even though it's the same basic mechanism, nevertheless, we are beginning to see differential behavior. That's what we mean by an emergent property. And so we're going to try to simplify these more complicated mechanisms by just dialing it back to the original mechanism, which is just simply catching and tossing a ball. Right? You can see some extraordinarily complicated behaviors here. Again, the only variable here is when we add a ball, the complexity gets bigger. We add another ball, the complexity gets bigger, and so forth and so forth. An example of a need to know for this class are the word roots for physiology. These really are a need to know. That is to say, if you see a new term and you don't know exactly what it means, look at the word root and understand what the word root is, and it'll give you insight as to what the word means. This is very much a terminology-driven course, so let's learn the word roots. Like, for instance, myo. Anytime we see myo, we think muscle. If we see poly, we see many, and so forth. Glucose can be carbohydrate or a sugar of some kind. Learn these because we will be using derivations of these words all semester, and having a conversational ability is one of the best ways that you can be successful in this course. Just one more here as I look at it right here. You see ACE, ASE. That means it's an enzyme. If you see lipase or ATPase, that means it's an enzyme that is doing something with whatever the rest of the word is. In that case, it's an enzyme involved with ATP of some kind, etc. So get to know these. These are really important, and they will serve you throughout your caregiving and clinical careers. Since I've already talked about homeostasis in some previous slides, I won't belabor those points any more than we have to. Again, we're always talking about our mechanisms with regard to a homeostatic window. Either we're trying to bring it back into a range that we want to be stable at, or in some cases, like I said, there are appropriate times to depart from that homeostatic window in order to meet a larger physiologic or reproductive need. So yes, we are keeping generally the internal environment stable, and that means that we have to perceive it. We have to link that perception with what I would call a doer, something that will get the job done, that we call an effector. So when we talk about cell signaling, we're going to talk about receptors, we're going to talk about secondary messages, and then effectors, things that get the job done in response. Sometimes a given molecule will do more than one role in a given system, but oftentimes we will see the sensor, integrator, and effector. Whether we're talking about reflexes or at the macro level, or we're talking about biochemical processes at the cellular level. We are also, again, referring to a homeostatic window, whether we're bringing it back into that window or pushing it away from that window. Here's a critical example of what we mean by homeostasis. pH. Now, many people will come into this class having forgotten what pH means. pH means that when it's the concentration of protons in solutions, hydronium ions and so forth. We'll talk about that, in class. In any case, it's the concentration of these, and the more you have, the lower the number. Okay? So when we are acidotic or we are in acidosis, that means the number of the pH is lower than what it should be. Battery acid is a pH of 1. Orange juice and lemon juice and so forth, pHs of 2 and 3, somewhere around there. On the other hand, if we are alkalotic, that means that the concentration of hydrogen is lower and the number is higher. And I'll give you the reasons why behind this. pH is basically a shorthand for concentration, and the lower the number, the more concentrated it is. The higher number, the less concentrated it is. A lot of folks come in and they have forgotten about pH. Make sure you get that locked down because it will just make your life a lot easier if you just know it instinctively rather than having to try to relearn it again. Relearn it now and keep it with you throughout the semester. Your life will be a lot easier, I promise. So let's take this one step further. We've already talked about the idea of changes, talked about homeostatic set point. All right, we have the receptors, we have the integrators, we attempt to correct. Sometimes we, though, are pushing away from a homeostatic set point if there's a particular physiologic need. But let's get at something that we talk about in terms of equilibrium versus steady state. Steady state and equilibrium are not the same thing. They are not the same thing. Sometimes we will achieve equilibrium, but oftentimes the body is working to maintain disequilibrium. Here's a simple example: temperature. If we were to achieve equilibrium with our environment, that would mean we would assume room temperature. And that's clearly not in our best interest physiologically. We would basically be dead. The fact that you breathe is meant to maintain disequilibrium so the gases flow in the right directions. All right? That we have oxygen moving into the bloodstream and carbon dioxide moving out of the bloodstream. We maintain a disequilibrium in order to achieve physiologic and homeostatic goals. That's why it's a steady state and not the same as an equilibrium. Given what we said in the previous slide, the idea that we need to maintain stability, a dynamic steady state, how do we do that? Well, it's expensive. It needs energy and it needs information flow. So a lot of Unit 1 is going to deal with the energy and the information flow. So let's talk about the energy side of it here for just a second. So you guys remember mitochondria, right? We're going to talk a little bit about mitochondria here in Unit 1 with the idea that we are generating ATP, right? We're integrating chemistry in order to govern the physics, if you will. So mitochondria are important for ATP, but mitochondria are also important for body temperature. Mitochondria change their function in order to meet metabolic needs, so we're going to adjust. And again, this is expensive. You might not realize it, but even if you only weigh 120 or 130 pounds every day, your body metabolizes over 150 pounds of ATP just in order to maintain disequilibrium across plasma membranes. This is expensive. That's a lot of ATP. Now, that's why the reason you don't weigh 150 pounds is the fact that you're recycling the ATP. So you have a given amount of ATP and you keep shifting back and forth between ADP or other forms, and then we convert it back into ATP. It's expensive. So we're going to talk about some of these processes and how we get the job done. Compartmentation, membrane functions, as well as some basic biochemistry. How these cells get the job done. And as mentioned before, one of the goals is disequilibrium. For instance, if we look at a cell here, we have the fluid inside the cell. We call that intracellular fluid. And we have the fluid outside the cell, which includes blood plasma, includes interstitial fluid, and so forth, the extracellular fluid. And these are not the same. And the body works very, very hard in order to sustain a disequilibrium across these two compartments. And what is the nature of that disequilibrium? Well, in the intracellular fluid over here on the right, and this is very much a need to know. We're going to talk about this literally almost every day this semester. This disequilibrium, this steady state between these two compartments. The intracellular fluid compartment, you can see, has relatively low levels of sodium ions and chloride ions. There are some exceptions. We will talk about them at the appropriate times, but for most cells, the vast majority of cells, we have very, very low sodium and chloride inside the cell. On the other hand, we have boatloads of potassium inside the cell. So we have lots of potassium and very little sodium and chloride. On the other hand, extracellular fluid is the opposite. We're quite low in potassium outside the cell, but we have lots and lots of sodium and chloride ions outside the cell. You remember in the previous slide I talked about how we metabolize over 150 pounds of ATP every day. We've been talking now for about half an hour. You have already metabolized on the order of somewhere around 20 pounds of ATP, and most of that ATP is sustaining this disequilibrium across the plasma membrane. So they're in steady state where we're sustaining these differences, but they are not at equilibrium. So investing all of this energy, what good or what are we achieving? What is it that we're really doing here? I've been lecturing for about half an hour now, which means that as you have been listening and learning about the themes here in chapter 1, you have metabolized somewhere around 10 pounds of ATP, most of which is sustaining the disequilibrium across the plasma membrane of sodium and potassium. Yes, chloride's in the mix in there, but chloride is not nearly as prevalent as sodium and potassium. We are literally going to talk about sodium and potassium disequilibrium almost every day this semester. Chloride has its place, and we'll bring chloride into the picture at some point. But for now, sodium and potassium are the big guns. And like I said, you've invested about 10 plus pounds of ATP doing nothing but sustaining the disequilibrium. Pumping, transporting is another word for this, sodium out of the cell and potassium in. And this enzyme here called the sodium potassium ATPase, notice that it says pump over here, and that's okay, but we want to use proper terms. So, maybe in a future lecture, I'll edit this out and make it the sodium potassium ATPase. You can remember that it's a pump, that it's moving things using ATP, but when it comes to exam answers, you're going to want to use proper terminology. So we are moving sodium out and potassium in using the sodium potassium ATPase. And again, what is the point? Well, if we differentially allow potassium and sodium to move, so we set up these gradients, and now sodium wants to diffuse back into the cell, and potassium wants to diffuse out of the cell, we now have a basis for things like voltage. Depending on how we let sodium and potassium move across the membrane, that changes the physical properties of the membrane in terms of the voltage on one side versus the other, which is the basis for signaling. For instance, maybe we let some sodium into the cell. That would mean this is a positive charge. It makes the inside of the cell more positive, which means that we can now have some kind of a signaling process. Or we can use the disequilibrium to do something else. We can say we've got lots of sodium outside the cell and it wants to diffuse in. Well, that's a source of energy. So maybe we have a transporter here that will move waste, which normally wouldn't necessarily move across the plasma membrane. Now we can move it by using the energy stored in that disequilibrium gradient. Again, we're not going to just leave that here. We're going to be seeing these processes again and again this semester. So I'm just kind of giving you an overview of what we're going to be dealing with here in Unit 1, talking about membranes, talking about diffusion, talking about gradients, and so forth to set us up for actual function. I'm going to stop right here for now since we've been going for a little while. We will conclude this chapter 1 discussion in the second video. Talk to you soon.